In this lecture, we will discuss the bacterial cell wall, which is present between the cell membrane and capsule of a typical bacterial cell and is very important in providing shape and rigidity to the bacterial cell structure. We will talk about the functions of the cell wall, the structure of the bacterial cell wall in detail, and classification of bacteria on the basis of cell wall structure. Let us first discuss the functions of the bacterial cell wall. The first function is maintaining the cell's characteristic shape. The rigid cell wall compensates for the flexibility of the phospholipid membrane and keeps the cell from assuming a spherical shape. The second function is countering the effects of osmotic pressure. The strength of the wall is responsible for keeping the cell from bursting when the intracellular osmolarity is much greater than the extracellular teichoic osmolarity. Yet another function is providing attachment sites for bacteriophages. Teichoic acids attached to the outer surface of the wall are like landing pads for viruses that infect the bacteria. And finally, providing a rigid platform for surface appendages. Flagella, fimbriae, and pili all emanate from the wall and extend beyond it. Bacteria have been classified on the basis of the structural differences in their cell walls and their reaction to a differential staining protocol called gram staining. These classes of bacteria are gram-positive bacteria, gram-negative bacteria, acid-fast bacteria. We will first discuss gram-positive and gram-negative cell walls in detail and later talk about the acid-fast cell wall. In 1884, a Danish physician named Hans Christian Gram discovered a differential staining protocol used to classify bacteria. Do you remember the Gram staining protocol? Well, let us quickly revise it. Prepare and heat fix the smear. Flood the smear with crystal violet for 1 minute. Pour off excess dye and wash gently in tap water and drain the slide against a paper towel. Expose the smears to grams iodine for 1 minute by washing with iodine, then adding more iodine and leaving it on the smear until the minute is over. Wash with tap water and drain carefully. Wash with 95% alcohol for 30 seconds. Wash with tap water at the end of the 30 seconds to stop the decolorization. Drain. Counterstain with saffron in for 30 seconds. Wash, drain, blot and examine under the oil immersion objective. The bacteria which retain the primary stain and appear violet are called gram-positive bacteria, and bacteria that take up the counter stain and appear pink are called gram-negative bacteria. These differences in the gram character is because of the differences in the cell wall structures of gram-positive as well as gram-negative bacteria. The cell walls of both gram-positive as well as gram-negative bacteria contain a complex macromolecule called peptidoglycan. Peptidoglycan, also called murine, is made up of alternating amino sugars N-acetylglucosamine and N-acetylmuramic acid polymerized by beta-1,4-glycosidic linkage. At the carboxy terminal of N-acetylmuramic acid, the lactyl group is substituted by a short tetrapeptide consisting of L and D amino acids. The peptide in the case of gram positives is usually made of L-alanine, D-glutamine, L-lysine and D-alanine. In the case of gram negatives, the tetrapeptide is made up of L-alanine, D-glutamine, mesodiaminopimelic acid and D-alanine. The presence of D-amino acids is known to protect the peptidoglycan from the attack of many peptidases. In the case of gram-negative peptidoglycan, a peptide bond is formed between the terminal carboxyl group of D-alanine present at position 4 of one peptide moiety and the non-alpha group amino of the diamino acid present at position 3 of an adjacent peptide. In many gram-positive bacteria, the cross-linkage is not direct but brought about by an additional pentaglycine peptide inserted between D-alanine of one peptide and L-lysine of another peptide. Thus, peptidoglycan is a complex meshwork of alternating N-acetylglucosamine and N-acetylmuramic acid residues with tetrapeptides hanging from N-acetylmuramic acid and cross-linkages between the adjacent tetrapeptides. The N-acetylmuramic acid residue represents the reducing end. The beta-1,4 linkage is susceptible to the attack by the enzyme lysozyme. Let us see a brief comparison of the gram-positive as well as gram-negative cell walls. The gram-positive cell wall structure, which surrounds the plasma membrane, is made up of a very thick layer of peptidoglycan. The gram-negative cell wall structure, on the other hand, is very complex with a thin layer of peptidoglycan surrounded by another layer called the outer membrane. The outer membrane is very important functionally. Because of its semi-permeable nature, it exhibits a strikingly reduced permeability to a wide variety of lipophilic compounds including bile salts and other detergents, lipophilic antibiotics and dyes. The space between the plasma membrane and cell wall is called the periplasmic space. The space is considered to be very active, containing enzymes for assembly of cell wall and membrane components, various degradative or deoxifying enzymes, secretion systems, sensing proteins for chemotaxis and signal transduction, and binding proteins for solutes taken up by transport systems. Let us now discuss the gram-positive cell wall structure in detail. The gram-positive cell wall consists of numerous interconnecting layers of peptidoglycan. Chemically, 60 to 90% of the gram-positive cell wall is peptidoglycan. Interwoven in the cell wall of gram-positive bacteria are teichoic acids. Teichoic acids are glycerol or ribitol phosphates and are linked by a phosphodiester bond to the carbon 6 of the muramic acid. Some teichoic acids have lipids attached and are called lipoteichoic acids. When bacteria are subjected to phosphate-limiting growth conditions, teichuronic acids which lack the repeating phosphate groups are synthesized. Teichoic acids have an affinity for magnesium ions and probably function as ion exchangers. Teichoic acids provide rigidity to the cell wall and also assist in regulation of cell growth by limiting the ability of autolysins to break the beta-1,4 bond between N-acetylglucosamine and N-acetylmuramic acid. Teichoic acids serve as an attachment site for some bacteriophages. They are also known to have endotoxin properties. Destruction of bacteria and release of teichoic acid into the bloodstream may cause fever, blood vessel dilation, and possibly shock and subsequent death. The gram-negative cell wall structure is more complex as compared to the gram-positive cell wall. As you can see, it consists of many layers, mainly a thin peptidoglycan layer, periplasm, and an outer membrane. The outer membrane is mainly a lipid bilayer, about 7 nanometers thick. It is composed of lipopolysaccharides, phospholipids, lipoproteins, and proteins. The outer membrane is highly asymmetric with an inner leaflet consisting of phospholipids and an outer leaflet of lipopolysaccharides. Typically, the outer membrane contains pore-forming proteins called porins necessary for transport of nutrients. Let us now discuss the structure of lipopolysaccharides. Lipopolysaccharide is a unique and major constituent of the outer membrane and is responsible for the endotoxic properties of gram-negative bacteria. Lipopolysaccharide of enteric bacteria consists of three structural regions: lipid a unique glycolipid anchoring the molecule into the outer membrane, a core oligosaccharide region, and peripheral O antigen side chains. The latter two extend out from the cell. Lipid A is highly conserved among gram-negative bacteria. It consists of a fatty acylated phosphorylated glucosamine disaccharide characterized by N-linked and O-linked 3-hydroxymyristoyl residues as well as saturated fatty acyl substitutions on the 3-hydroxy fatty acids. The inner core backbone region consists 2,3-L-glycero-D-manoheptose residues and the outer core oligosaccharide is glycosidically linked to the non-reducing heptose residue. The O antigen consists of long polysaccharides generally composed of oligosaccharide repeating units and is antigenic in nature. Let us also discuss the functions of lipopolysaccharides. The first function is it contributes to the negative charge on the cell surface. The second function helps stabilize outer membrane in a manner similar to the glycopeptides and teichoic acids of the gram-positive cell wall. Creates a permeability barrier. It is responsible for the strikingly reduced permeability to a wide variety of lipophilic compounds including bile salts and other detergents, lipophilic antibiotics and dyes. Protects from host defenses. Role in pathogenesis. The outer membrane also plays a crucial role in many aspects of pathogenesis, including the evasion of host defenses and adherence of invading bacteria to host surfaces. Let us now discuss the third type of cell wall, the acid-fast cell wall. They are called acid-fast because when they are stained with a basic dye like basic friction, they resist decolorization with either dilute acid or acid alcohol and retain the primary stain and hence the name acid-fast. On counterstaining with methylene blue, The acid-fast bacteria appear red, whereas non-acid-fast bacteria appear blue. The acid fastness is due to its cell wall structure. Common acid-fast bacteria of medical importance include Mycobacterium tuberculosis, Mycobacterium leprae, and Nocardia species. Acid-fast bacteria are gram-positive, but in addition to peptidoglycan, the outer membrane of acid-fast cell wall contains large amounts of glycolipids, especially mycolic acids. The acid-fast cell wall of Mycobacterium has a thin inner layer of peptidoglycan. The peptidoglycan is in turn linked to arabinogalactan, which is then linked to an outer membrane containing high molecular weight mycolic acids. The arabinogalactan mycolic acid layer is overlaid with a layer of polypeptides and mycolic acids consisting of free lipids, glycolipids, and peptidoglycolipids. Other glycolipids include lipoarabinomannan and phosphatidylinositol monocytes. Like the outer membrane of gram-negative cell wall, porins are required to transport small hydrophilic molecules through the outer membrane of the acid-fast cell wall. The outer surface of the acid-fast cell wall is studded with surface proteins that differ with the strain and species of the bacterium. The periplasm is present between the peptidoglycan and the cytoplasmic membrane. Let us now discuss the functions and advantages of the acid-fast cell wall. The mycolic acids and other glycolipids impede the entry of chemicals, causing the organism to grow slowly and be more resistant to chemical agents and lysosomal components of phagocytes than most bacteria. There are much less porins in the acid-fast cell wall as compared to the gram-negative cell wall, and the pores are much longer. This is thought to contribute significantly to the lower permeability of the acid-fast bacteria. The surface proteins in the acid-fast cell wall, depending on the strain and species, carry out a variety of activities including functioning as enzymes and serving as adhesins. Adhesins enable the bacterium to adhere intimately to host cells and other surfaces in order to colonize and resist flushing. Having studied all the three cell walls now, let us now discuss and compare the three types of cell walls that we have just studied. If we consider the thickness of the cell walls, the gram-positive cell wall is very thick, about 20 to 80 nanometers thick. Gram-negative cell wall is much thinner with 10 nanometer thickness, and acid-fast cell wall is also considered to be thick. The second important function is the outer membrane, which is absent in gram-positive cell wall and present in both gram-negative as well as acid-fast cell walls, though the composition is different. As far as peptidoglycan content is concerned, it is very high with more than 50% in gram-positives and lower with around 10 to 20% in both gram-negative as well as acid-fast cell walls. The teichoic acids are a characteristic feature of gram-positive cell wall, whereas they are absent in gram-negative and acid-fast cell walls. The lipid content in gram-positive cell wall is very less, whereas very high in gram-negative cell wall in the form of lipopolysaccharides and mycolic acids in the case of acid-fast cell wall. The periplasmic space is generally absent in gram-positive cell wall and acid-fast cell wall, but present in gram-negative cell wall and harbors many enzymes and other molecules, as we have already studied. The presence of these different types of cell walls impart a rigid shape to gram-positive cell wall, and generally speaking can be either rigid or flexible in the case of gram-negative and acid-fast cell walls. As a general rule, gram-positive cell walls are very sensitive to antibiotics. Gram-negative cell walls are moderately sensitive and acid-fast cell walls are least sensitive. Likewise, gram-positive cell walls are sensitive to enzymes like lysozyme, which attacks the beta-1,4 linkage between N-acetylglucosamine and N-acetylmuramic acid, whereas both gram-negative as well as acid-fast cell walls are not sensitive to lysozyme. Let us now discuss some frequently asked questions in the university examinations. First, we will discuss short answers which can be asked for 2.5 or 5 marks. Describe the principle of gram staining. Yet another question may be, write a short note on peptidoglycan structure. Third question can be, write a short note on lipopolysaccharide structure. A question which is asked almost every year is, distinguish between gram-positive and gram-negative cell wall structures. Coming to the long answers which can be asked for 10 marks. The first question is, with the help of a neat labeled diagram, describe in detail the gram-negative cell wall structure. Another frequently asked question is, describe the acid-fast cell wall structure. Also, discuss the advantages of acid-fast bacteria over other bacteria.